Reprogenetics is the use of reproductive and genetic technologies to select and genetically modify embryos with germinal choice technology for the purpose of human enhancement. However, a report by the Hastings Center broadly defined reprogenetics as any creation, use, manipulation, or storage of gametes or embryos. The term was coined by Lee M. Silver, a professor of molecular biology at Princeton University, in his 1997 book Remaking Eden. Definition. In Silver's formulation, reprogenetics will involve advances in a number of technologies not yet achieved, but not inherently impossible. Among these are improvements in interpreting the effects of different expressions of DNA, the ability to harvest large numbers of embryos from females, and a far higher rate of reinsertion of embryos into host mothers. The end result, according to Silver, is that those parents who can afford it will be able to pick out the genetic characteristics of their own children, which Silver says will trigger a number of social changes in the decades after its implementation. Possible early applications, however, might be closer to eliminating disease genes passed on to children. According to Silver, the main differences between reprogenetics and eugenics: the belief in the possibility of improving the gene pool, which in the first half of the 20th century became infamous for the brutal policies it inspired, is that most eugenics programs were compulsory programs imposed upon citizens by governments trying to enact an ultimate goal. Reprogenetics, by contrast, would be pursued by individual parents who would be trying to improve their children with the same motivations that compelled them to purchase expensive courses in preparation for standardized testing, e.g., the SAT. Eugenics would have required a continual selection for breeding of the fit and a culling of the unfit, while, according to bioethicist James Hughes, universal access to reprogenetics provided by a welfare state would permit the conversion of all the unfit to the highest genetic level. However, he shares Silver's concern that unequal access to reprogenetics could create a two-tiered society of gen-rich and gen-poor, genetically engineered haves and have-nots. See the film Gattaca for a fictional depiction of the latter scenario. Towards the end of Silver's book, he speculates that the gen-rich and the naturals could, over time, even become separate species, unable to interbreed. However, Silver now accepts the criticism made by many evolutionary biologists that speciation cannot occur without strict reproductive isolation and is therefore extremely unlikely to happen. The other contrast is that it is now known that the concept of genetic purity through eugenics is misguided. This form of genetic purity, insofar as it is meaningful, is effectively inbreeding and results in poor health and infertility, while the end result of reprogenetics on the gene pool would be reduced incidence of genetic disease and potentially increased genetic IQ reception. Skeptics think the goal of using reprogenetics to increase genetic IQ is rendered less likely by the very great complexity of the genome with respect to intelligence. Half of all genes are expressed somewhere in the brain, not to mention the environmental influences from conception to adulthood. Although research in the area of the inheritance of intelligence is still fairly contentious, these findings and others accumulating evidence give support to the idea that there is a finite number of genes that determine general intelligence, and not just separate genes determining individual intellectual capacities such as memory, spatial visualization, or verbal skills. Some argue that genetic disease will never be eliminated, since mutations and chromosomal errors, e.g., Down syndrome, will always arise. However, some bioethicists, such as Julian Savulescu, argue that this fact would not alter the existence of a societal and parental obligation to provide genetic and non-genetic health to children. Also, prenatal screening techniques are not limited to reprogenetic methods only capable of detecting inherited conditions. Combined with tests such as those currently used to screen embryos harboring Down syndrome, spina bifida and similar pathologies, both inherited and incidental genetic disorders can theoretically be prevented from reaching the phenotype stage. More importantly, critics think that heterozygote advantage means that elimination of a genetic disease might lower the fitness of the majority or at least a significant number of people, well-meaning though it might be. Silver counters that this argument assumes that all members of a species function together in genetic terms. According to Silver, this assumption has no basis in reality, since the concept of a gene pool was invented as a tool for developing mathematical models by biologists who study populations of animals or plants. It is calculated as the frequencies with which particular alleles of particular genes occur across all of the members of a population that interbreed with each other. See also